Hello everyone, in this video I'm going to be talking about something called phagocytosis. Now this is typically taught at A level and IB when you think about defence mechanisms and the way the body responds to infection. Phagocytosis is a non-specific response to infection. Now I do a separate video on the difference between non-specific and specific responses, because specific responses are those that initiate what we know as B cells and T cells, our big white blood cells. Phagocytosis, however, is a response that we have, and it's the same for all pathogens. It's a non-specific response to a pathogen, to an infection, let's say. So what I've got here are just a few bits, and then I'm going to explain the process a bit more. So the breakdown of large particles like bacteria by white blood cells, and this is the key bit, these blood cells that we're talking about in this video are called phagocytes, hence the name. So phagocyte is the white blood cell, phagocytosis is the name of the process. So they're engulfed by these cells, these phagocytes, in the form of vesicles, which I'll draw out from the cell membrane. So if you're familiar with pac-man at all, it's a little bit like that if you were to think about it in general terms. Now if we did have an infection, ideally you want as many phagocytes getting to that site of infection as quickly as possible. So the body responds by releasing histamine. So we release histamine, and that dilates the blood vessels, and it will allow more of these phagocytes to get to that site of infection. Now that's what we know as inflammation. So when we get inflammation, that's what's happening, and you get a collection of pus forming at that site of infection. Now pus actually is just dead pathogen, or dead pathogen and organism causing disease. So dead bacterial cells and phagocytes collectively we call pus, and that starts to form at that site of inflammation. So in phagocytosis we're basically just breaking the bacteria down using the phagocyte cell to do it. So what I'm going to do is draw out as best as I can what happens and explain the process as we go along. So I'm going to draw the outline first of something, and then I'll explain exactly what it is that I'm drawing. So it's going to look a little bit odd perhaps at first, but so what we've got here, no, it's not the best sketch, but what I've drawn here is a phagocyte. A phagocyte, and all these little bits that I've just drawn jutting off this phagocyte, these are called pseudopods. Now I could have drawn a simplified version of this picture, but I think it's important, if I can shade all of this area in here, I think it's important to show it as realistically as possible. So this is my attempt at trying to draw three-dimensional looking almost diagrams. I thought it's time to upgrade the pictures that I've been drawing. So here we have a massive cell, a phagocyte. So this whole picture is of one phagocyte, and each of those little yellow bits that you can see jutting out are called pseudopods. And this whole area inside the middle, so this part here, if we just label this in red just to make it clear, this part here is cytoplasm within that cell. So the first thing that happens is, well, we need to put in place our bacterial cell. So for the sake of argument, this blue circle here is going to represent our pathogen, the organism that's going to cause disease, which is our bacterial cell. So that blue dot that I've drawn is going to be our microbe, which in this instance is a bacteria, and it's going to come and it's going to move a bit like that. What you have is something called chemotaxis and adherence of the microbe to the phagocyte. So chemotaxis, chemo means chemical and taxis is movement. What you find is that the chemical products of the pathogen, so this pathogen, this microbe, will actually release chemicals that oddly attract the phagocyte. So it inadvertently releases chemicals that will attract that phagocyte, and you get what's called chemotaxis, chemical movement. So the phagocyte knows exactly where to target. So you get chemotaxis. So this is stage 1 here, and I'll make a list of, or a short summary over on here on the side. So you get what's called chemotaxis and adherence. Adherence is in the microbe starts to stick to the phagocyte. Now the reason why I've drawn these parts here, what appear to be, I'll just shade them in, enveloping or engulfing, if you like, that microbe. That's what I was trying to refer to by my whole pac-man thing. This part of the membrane, if you like, surrounds and completely engulfs that microbe. And that's number 2. So for number 2, what you get is ingestion of the microbe by the phagocyte. So that's called ingestion. You envelop, with that cell membrane, you envelop that microbe, you wrap around it. And if you continued, what that would form would be something like this. It looks a bit unusual. But you'd have the microbe contained within its own sort of little membrane sac. Its own little vesicle, if you like. A vesicle is a membrane-bound sac, and that's why this is something called a phagocytic vesicle. So what we form at number 3, if we imagine that microbe being engulfed or enveloped by the membrane, you get the formation, so number 3 is the formation of what's called a phagosome. So we have the formation of a phagosome. Then in the cell, you might be aware from previous videos on cell structures, you have something called a lysosome. This is an organelle, a part of the cell that you find, and the lysosome contains digestive enzymes. And that lysosome oddly goes to fuse with the phagosome. And it starts to form, and it's a combination of the two names, so phagosome with the lysosome, that fusion forms what's called a phagolysosome. And I'll add it here, phagolysosome. So at number 4, here, you get a fusion of the lysosome to the phagosome to get a phagolysosome. And you can imagine that if the lysosome contains digestive enzymes, then you're going to ultimately digest that microbe. So those enzymes, as you can guess, start to digest and break down that microbe. Now, there will be bits of indigestible material. The enzymes won't be able to break down all of it, and that indigestible material remains in what's called a residual body. So once you get the formation of the phagolysosome, there'll be little bits that the enzymes can't digest, and they remain in what's called a residual body. So you have a residual body of indigestible material. And that's really our sort of 5th stage. Now you're clearly not going to want a residual body remaining in the cell. So what we're going to simply do is try and discharge it We're going to discharge all that waste, and we're going to get rid out of our cell of all that indigestible material. And that's what the final step is. So what we do in our final step, take that residual body, and via exocytosis we discharge all that waste. And that's the final step there. And that ultimately is phagocytosis. So you get chemotaxis and adherence of the microbe to the phagocyte, number one. And number two, we get ingestion of the microbe by the phagocyte. Number three, we get the formation of this vesicle called the phagosome. The phagosome joins with a lysosome containing digestive enzymes to form a phagolysosome, and that's number four. Those digestive enzymes break down that microbe, and any undigested material remains as a residual body, that's number five, and then we discharge all that waste material, and that's point 6 on the diagram. And that ultimately is our process of phagocytosis. And when we're infected by things like bacteria, we get as many of these phagocytes as we can to get to that site of infection to destroy that bacteria. Okay, hope all that helps.